Hey everyone, in this video I'm going to be jumping into bacteria as pathogens, and I'm going to be going down this roller coaster of research into bacteria as pathogens, how they cause disease, how we treat bacteria, and how we might be using AI to design the next generation of drugs to kill these bacteria. So let's jump into it. Here we have a beautiful forest of bacteria. Now this is actually pseudo color because they would have had to have used an electron microscope which doesn't have color to create this image. So they take the electron microscope image and then they color it sort of depending on what species of bacteria they see, but they can certainly end up with some beautiful images like that. So let's jump into it. I can't cover all the bacteria. There's millions of bacteria. So I'm going to be covering these main bacteria, and these are important for various reasons. So these are clinically important, but they're also across the spectrum of kinds of bacteria that we get exposed to, just to give you some case studies of important groups of bacteria that we come across. So we've got a coli and Staphylococcus aureus, which I'm sure you've heard of. We've got Pseudomonas, and we've got Clostridioides. They just changed the name from Clostridium to Clostridioides difficile, but everyone just calls it C. diff. So here we go. We've got a coli, Staph aureus, Pseudomonas, and C. diff. That's what I'm going to call it throughout the video, but that's their full names there. I'm terrible at Latin. I'm sorry. I'm a scientist, but I'm terrible at Latin. I never learned that kind of stuff. All right, here we go. So first of all, how do we break these bacteria up? Now, we always break these bacteria up by gram-negative and gram-positive. These are the two major groups of bacteria, and they have different membrane structures, which means that they stain differently under a gram stain. So gram-negative bacteria have two membranes and a small peptidoglycan layer, whereas gram-positive have one phospholipid membrane and a single large peptidoglycan layer. And it's the peptidoglycan that stains under gram-positive, which is why gram-positive are purple. They're staining positive for gram because of that very large peptidoglycan layer. So how do our bacteria break up? So we get E. coli and Pseudomonas as gram-negative, gram-positive is Staph aureus and C. diff. And so that now you can see I'm trying to cover the diversity of bacteria that you might see and group them up nicely. So another way to break them up is by shape. And actually these are kind of boring. Most of them are bacillus, which means rod-like shape. Some of them can flip between bacillus and diplobacilli, which basically means two bacillus stuck together. For example, Pseudomonas can do that. And then the next one is the bunch of grapes, which coccus means a circle, it means round. Staphylococcus means a bunch of grapes, a cluster of circles, and that's Staphylococcus aureus, obviously. Now, another way to break them up is to opportunistic pathogens or obligate pathogens. Now, this one, I haven't covered the diversity of pathogens here. These guys are all opportunistic pathogens. What does that mean? It means that they're not always disease causing. You might find them on you or in you and you don't have the disease, or you might find them in the soil, in the water, in your food. And so really some situation has to happen for them to transition from an opportunistic pathogen into an actual disease. So normally they can be okay, sometimes they turn into disease. Now, obligate pathogen means that at some point in time in their life they have to be inside a human and they have to be causing disease. So chlamydia is an example of this, where its life cycle is actually inside our cells. And so because it has to get into our cells and then it has to burst that cell to get out of the cell, you can see by its very existence, it's a pathogen. But there's kind of another way to say that, and that is if these bacteria are in us at a reasonable level, are they going to be pathogenic, benign, or mutualistic? And this, we can start to see some differences in this bacteria, in these bacteria. And at this point, I would start to say they break it up like this. I would go as far to say that you want E. coli and Staphylococcus aureus on you or in you. You want it, and that means that they are mutualistic. They benefit us and we benefit them. There is mutual benefit in this relationship. So I'd go as far to say that we want those in there. Why would we want them? Well, E. coli's in our intestines. It outcompetes potentially pathogenic bacteria, but also it produces vitamin K, which we absorb. Staphylococcus aureus can be in our lungs, and studies have shown that if it's in our lungs at low levels, it can actually outcompete more pathogenic bacteria, including Pseudomonas. Whereas Pseudomonas and C. diff, you don't ever want on you. And studies that show that they do appear up in healthy people, and sometimes they don't cause disease. The infections are normally transient. In other words, our body's probably trying to get rid of it. So, for example, around 1 in 30 people will have C. diff in their intestines and still be healthy. But if you were to test them 6 months later, a lot of those people wouldn't have it anymore. It was a transient, maybe they ate something that day or the day before that had C. diff, but it wasn't at a dose, at a concentration that would cause disease. But in general, if your body tested negative and completely for Pseudomonas and C. diff, you'd be a happy chappy. You'd be happy to not have those bacteria in it. Whereas, especially E. coli, you would want that to be in there. And so there are differences in these bacteria in terms of their pathogenicity and their mutualistic relationships with our body. All right, so what makes these 4 opportunistic pathogens become pathogenic? And there's actually a number of things. So I'm going to go cover a list of things here. So number 1 is dose. Yes, so dose affects whether it becomes a pathogen or not. And so my example for this, I found a great study on these things called rice cakes, which is a Korean treat. It's got a bean paste, it's got a lot of sugar, it's rice, and it's wrapped in sesame seed, and it's apparently delicious, and it's often served at room temperature. Now, these rice cakes grow Staphylococcus aureus at quite a substantial rate. And so basically for these experiments, they generated the rice balls and then they watched the growth curves of Staphylococcus aureus in these rice balls, in these rice cakes. And so we've got 20 degrees, 30 degrees, and 40 degrees in times an hour. So you can see after 1 day, 24 hours, we're pretty much maxed out on Staphylococcus aureus in these samples. That is a very high dose of Staphylococcus aureus. So if you were to eat these rice cakes, if they had been room temperature for 1 day, you would get very sick. So in fact, it's recommended that you don't want more than 10 to the power of 4 Staphylococcus aureus in a food that you eat. So that's right there. So you can see very quickly within 12 hours at 20 degrees, which is a cool room, you are going to have a potentially pathogenic level of bacteria. Essentially, at that concentration, if you have such a high concentration, it can overwhelm your immune system, but also it can start to form things like biofilms, which I've covered previously, which means that the bacteria can now curate their environment to be a better environment for them to divide and evade the immune system and to break down and feast on our cells, which is what Staphylococcus aureus does. It produces a toxin, for example, called hemolysin, which pops our cells so it can feast on its innards. So yes, dose is critical to whether a pathogen becomes opportunistic or whether it's healthy. If we take a large dose of Staphylococcus aureus, it can transition from opportunistic to pathogenic. What I loved about this paper is they actually got this saying is filled with a delicious sweet red bean paste. So now it is scientifically published that these are delicious. It's not a debate anymore. It's published in the peer-reviewed literature that these things are delicious. And I love that the authors snuck those in to the publication. Anyway, so they put the rice balls in the fridge at 5 degrees and in a cool room, 10 degrees. 10 degrees a little bit warmer than a fridge. And these bacteria didn't divide at all. In fact, they went down in the 5-degree treatment. So it was actually too cold for the Staphylococcus aureus. And this is why we refrigerate our food. And this is why you should refrigerate these rice cakes and also probably sushi. Don't eat room temperature sushi unless it's just been made fresh, because rice is a perfect growing medium for things like Staphylococcus aureus. Interestingly, Pseudomonas is happy to divide at 4 degrees. So it'll divide and divide and divide at 4 degrees, unlike Staphylococcus aureus. So you often find Pseudomonas in your fridge, in your foods on your fridge. Okay, the next big factor on whether something is opportunistic or pathogenic is location. Now, you kind of intuitively know this. We have E. coli in our large intestine, and when we poop, 50% of the weight of that poo is bacteria, and that bacteria wasn't harming us when it was in our large intestine a second before we did our poop. But if we were to then, I'm sorry to say this, eat that poo, we would get very sick. And all we've done is changed the location. That bacteria was in our large intestine, but now we've put it down our mouth, we've changed the location, and now it's going to make us very, very sick. So location is critical to whether an opportunistic pathogen becomes pathogenic. Essentially, our body has evolved to deal with bacteria in certain locations, and when bacteria go into different locations, that's when they can start to cause a problem. So, for example, in healthy humans, around about 0.1% of all bacteria in your large intestine are E. coli. However, 90% of urinary tract infections are due to fecal contamination. There are fecal E. coli are now in your urethra causing a urinary tract infection. And so essentially we can see that there's been this translocation. Essentially fecal bacteria have now got up the urethra and caused a urinary tract infection. And you know, this is a very important thing, particularly with young babies with nappies, and you get poo around the urethra, particularly in females because they have a smaller urethra. Biological females have a smaller urethra compared to males, and so there's less distance for the bacteria to get all the way up into the bladder and cause real problems. Okay, so yes, we've got contamination, but also E. coli can cause meningitis. Your meninges is the bag that contains the fluid that contains your brain. So the meninges are sort of that protective layer on the outside. Inflammation and infection of the meninges causes meningitis, very dangerous, squeeze of the brain, body temperature, not good. And E. coli can cause that. But how would E. coli get into your meninges? It must have done some crazy translocations or something to get there, perhaps even a brain injury, an external trauma that has broken the meninges and allowed bacteria in. Yeah, so location is critical. A healthy bacteria in one location can cause a disease in another. And just to break this down even more, Staphylococcus aureus is often in our nose. If I was to swab you and I wanted to find some Staphylococcus aureus in the nasal cavity, is a really good bet. Around 70 to 90% of healthy humans have Staphylococcus in their nasal cavity. But interestingly, there is this pathway from nasal cavity down to your wounds. So it's in your nasal cavity, but then you touch your nose and now it's on your fingers. Maybe it's a little bit runny, or some boogers, maybe you're plucking some hairs. Now you've got it on your fingers, and then you get a puncture wound on your skin in a region that doesn't normally contain Staphylococcus aureus, say like the sole of your foot. And then you touch the sore. You're like, oh no, I've got a sore there, and you touch it or you pick the scab. You've now implanted some Staphylococcus aureus into a wound that was up your nose. And so it's gone from a healthy location to inside a wound. You've broken the barrier with that nail. So now it's spread and it can feast on the cells of the foot, causing giant ulcers, and it can spread and eventually become septic. Now, there was this wonderful study that showed that 80% of Staphylococcus aureus skin lesions, when they did the genetic matching of the bacteria, it matched the bacteria up their nose. And so the DNA, the exact DNA scripts of the Staphylococcus aureus in their ulcer was in their nose. So it suggested that there was a translocation of that bacteria. So again, location. And I'm going to break down location even more because we've got two more of environmental bacteria. These are often found in various locations like meat, water, tap water, which is a bit of a problem. Around 3% of bottled water contains Pseudomonas, which is terrifying, but much higher percentage of tap water, for example, and it's also in the soil as well. So around about 35% of meats in the sample, this was a Spanish study, contains Pseudomonas. But C. diff was found in 15% of puddled water. This one was in America, and 37% in the soil samples there. And there are the researchers there. So yes, these are environmental potential pathogens, opportunistic pathogens, and they're healthy. They're fine to be there. It's when they get into us at a high enough dose that things get in problems. But also another big location thing here is hospitals. Hospitals have a lot of bacteria in their environment. These bacteria can often be antibiotic resistant because they've been exposed to antibiotics, and they're being exposed to vulnerable patients. So we've got a lot of bad things going on. And these three bacteria are huge when it comes to hospitals. Now, there's nosocomial infections is the technical term, but people are starting to use better language now, so they're saying healthcare associated infections. So these three bacteria are very big in healthcare associated infections. And for example, they can become urinary tract infections when they're on the tubing that goes up into a catheter. 75% of urinary tract infections in hospitals are catheters, and it can be lethal. Trust me, it can be lethal, these urinary tract infections. And around 24% of these infections in one study were E. coli and 10% were Pseudomonas, and they were hiding in the tubing. And you might think, well, shouldn't you be disinfecting that tubing? Let me touch on that in a sec. Because another location is in respirators. Within these respirators, we find viral, yeast, as well as bacterial infections, and they can cause infection through these respiration issues. And roughly, when they've done some surveys, it's a little bit all over the show depending on the hospital, but after probing these machines, they found roughly 25% E. coli, 25% Staph aureus, and 25% Pseudomonas. Aeruginosa is the strain name there, species name there. Yeah, so these are very common infections in ventilators and people who are on ventilators, and they seem to come from the tubing itself. Now, in studies that actually looked at the tubing, so this study, what they did was they took tubes, they inoculated them with Pseudomonas, and then they immersed them in different kinds of disinfectant, and then they checked how much bacteria should we get. So here we have on the y-axis bacterial colony forming units, that's how much bacteria there is, and down on the x-axis we've got sterilizing agents. So water was the control, and then this was hydrogen peroxide, which we would normally consider a very good harsh sterilization, but it did almost nothing to the Pseudomonas. Bleach is very similar, and Clorox, which is a commercial hospital brand disinfectant, it reduced it by maybe 50-60%, although that is on a log scale, so a lot, but it didn't get rid of it entirely. And that's a real problem. And in fact, some studies have actually looked inside the disinfectant in hospitals in the bottle, and they have found Pseudomonas because it's that resistant to these disinfectants. So it's a very tricky thing to get out of your environment. And obviously, if it can get from that tubing into our body, that's a change in location, and those opportunistic bacteria can now become pathogenic. Next up is a weakened host or a vulnerable patient, another way to see that. Now, there's lots of examples of that, being old, being young, this kind of thing, post-COVID perhaps. But some big examples is HIV, which is a virus that attacks your T cells, which are the cells that coordinate your immune response largely. So without your T cells, you aren't going to be able to resist even a tiny dose of these opportunistic pathogens. Radiation therapy, so on the y-axis we've got white blood cells, those are your immune cells, and it's fold baseline. So what we can see is, 4 days after radiation therapy, your white blood cell count starts to decline dramatically. White blood cells are very sensitive because they're fast dividing to radiation. So a person going through radiation therapy for their cancer, very vulnerable to these opportunistic pathogens because they can't fight it with their immune system. Another thing that affects your immune system is a thing called bone marrow failure. There's a number of causes of this, from cancer to genetic hereditary conditions. And what we're looking at here is PET imaging, and this is a radio-labeled tag that tags an active, healthy bone marrow, essentially, stem cells in your bone marrow. And it's those stem cells that produce your immune system as well as your red blood cells. So in a healthy patient, we can kind of see the skeleton because we can see the bone marrow. So you can see the rib cage, the spine, the head of the femur there. That's a big area for bone marrow. But in a person with bone marrow failure, we can't see the skeleton because the radio-labeled ligand isn't binding to the active stem cells within the bone marrow. So this person's lost their bone marrow. It's a very terrible condition to get. And so they're very vulnerable to these opportunistic pathogens. The next one is dysbiosis. So dysbiosis is an imbalance in the bacterial colonies within your body. And so I'm just going to jump through a piece of research that I think is a very good example of this by Deng et al. And it very nicely explains the relationships between some of these bacteria. And in this one, they're looking at B. fragilis versus C. diff. You can kind of imagine it. So let me jump through the experiment paradigm. They take a mouse, then they give them multiple antibiotics, a lot of antibiotics, to kill off all the bacteria within this mice. Then they inoculate them with C. diff, giving them a C. diff infection. That's group 1. Group 2, they first inoculated with B. fragilis, then they gave them the C. diff infection. That's group 2. Group 3 is they gave the C. diff infection as well as the antibiotic, an antibiotic treatment that's known to be effective for C. diff. So this is sort of the positive control group. Group 3 is the positive control. Group 1 is the negative control. And group 2 is you want to see what happens. How does the B. fragilis affect the C. diff infection? So first, let's look at survival. Now, I don't know what country this is in, but many countries have banned survival studies. But here we have a survival study. And so basically, mice that weren't infected with C. diff survived. So you got 100% survival. Group 2, that's the B. fragilis group. It has a huge amount of survival there. Only a few mice have died, and we've got 85% survival. Group 3, they were given C. diff and the antibiotic treatment. They're looking at 65% survival throughout the study. And the C. diff infection group seemed to have killed most of the mice. I would guess that maybe that's one mouse left. So it killed 90% of the mice. I would say 9 out of 10 of the mice, I'm guessing, were killed by the C. diff infection. So it's a very fatal infection. But importantly, we can see that inoculation with B. fragilis prevented the C. diff from being lethal. Now, how did they do this? Well, actually, they started to break down the research to figure out how was B. fragilis preventing the C. diff from becoming pathogenic. Interesting question. So let's jump into it. First, it seemed to be competition. Merely by coating the surface of, so here we have an intestinal villi. By coating the surface of the villi, they outcompeted the C. diff, blocking them from entering the villi and getting access to the villi in order to cause pathogenicity. So first they outcompeted. So without the B. fragilis, the C. diff was free to fully digest and invade. Next up was mucus. Now, B. fragilis seemed to be releasing chemicals that induced the secretion of mucus by the host cells. So our cells normally pump out mucus, and that's normally protective in the intestine. The B. fragilis seemed to put that into hyperdrive with what they were secreting. So we end up with a very thick mucus layer, and that again prevented the C. diff from getting access to the intestinal villi. Now you can see a good evolutionary reason for this. The B. fragilis actually likes to live in the mucus, so it wants the host to produce the mucus because that is generating a great environment for itself, but it's also helping us to prevent the C. diff infection. So that's another way that it's happening. A third way is the B. fragilis seem to also promote the growth of a more healthy flora, a more greater diversity of bacteria within the intestine, and they too can outcompete the C. diff. So here we can see that they've promoted the growth of mutualistic bacteria, again blocking the C. diff from getting access to the villi. And here's just some of the data that supports what I've explained. So in green we have mucus, and over here we had the C. diff with B. fragilis. We've got a lot of mucus in these cells, and with C. diff alone we don't have a lot of that green fluorescent mucus there. So yes, we can see that we've got more mucus being produced in the presence of B. fragilis. Here we have adhesion rates. This was actually done in cell culture. They plated up some cells, they put the bacteria on it, and then they washed the cells with media and looked at how many bacteria stayed stuck to the human cells. So they had the human cells growing in the dish, they put the bacterial cells on, and then they looked at how well they stuck. So when they put just C. diff on, they got almost 95% adhering to the cells, and you couldn't wash them off. So they'd adhered fantastically, probably using Fimbriae and other virulence factors in order to adhere to the host cells. Whereas the B. fragilis and C. diff co-cultures showed almost no adherence. They've actually split the y-axis there, which kind of hid their point that, we're looking at 6%, so 94% reduction in adhesion when they included the B. fragilis. So they massively blocked the adhesion there. And lastly, here we have, they can do sequencing to see what sort of spectrum of bacteria you've got, from red to blue to green, and these all correspond to different family of bacteria, different clades of bacteria, I should say. So we can see in a healthy person, you've got a good diversity, a good range of colors there, and that diversity is what you would want and expect in a healthy person. In the C. diff inoculation, it looks like you've only got 2 clades of bacteria there. So you've you kind of got a monoculture, almost a dual culture. You've only got these two bacteria there. And then over here, with the C. diff, you can see you've gone back to that healthy diversity of bacteria. So they've promoted the growth of a range of bacteria. And that healthy diversity of bacteria is what you want to see in the gut. So this right here in a single picture is dysbiosis. This is what we're talking about here. So next up, we have virulence factors, but I'm conscious I've been going on for a little bit while. So I want to actually spend a whole video breaking down some of the bacterial virulence factors that allow an opportunistic bacteria become pathogenic. Thanks, Deng. So that'll be in the next video.